Thank you, Pawan, and thank you, Sunil and Amit, for the invitation into PHIMSA. So I'm going to share my screen, and hopefully you can all see it now. So, is this visible to you? Yeah. Okay. So today I'm broadly going to talk about approaches to human immunology. I was also asked to just broadly cover some overview of immunology, but I've noticed that you've had a very good coverage today of innate immunity, and there was probably a wonderful discussion on the thymus earlier in the day. So I'll just touch briefly as an overview, and I'll come back to it maybe in some other form at the end of the lecture. So we all know that we have innate and adaptive immunity. Innate immunity has barriers, all the cells you've been hearing about, complement, type 1 interferons, pro-inflammatory cytokines. But adaptive immunity is mainly mediated by B cells making antibodies and T cells, which can either help other cells or can be cytotoxic. Okay, and innate immunity is immediate, adaptive immunity is late. Innate immunity, we break into inflammation and the antiviral state. You can neatly divide it into these two halves. And you've heard a lot about inflammation today. Inflammation comes in different forms in disease. This is just acute inflammation that's depicted here. This is what we use when we think about inflammation soon after an infection. But if you're thinking about a chronic disease, there's more to it. So when you're studying human disease, you actually want to know what is the nature of the inflammation, what's driving it. What is the adaptive component that's triggering it if there is an adaptive component? And so this is one half of innate immunity. The other half of innate immunity is the antiviral state. This is, of course, very important in COVID-19. Now we realize that because the antiviral state depends on the generation of type 1 interferon. And tomorrow, when I talk about COVID-19 and some of our work, we'll briefly discuss how this is subverted by the virus. So type 1 interferons bathe other cells, signal through the type 1 interferon receptor, and induce in these recipient cells an antiviral state. And type 1 interferons also affect adaptive immunity in NK cells, and so on. Okay? Now, we think about the sentinel cells of innate immunity as being mast cells and macrophages, the early for inflammation. So mast cells and macrophages, and you heard all day about TLRs and the activation of these cells. And then we also have the dendritic cell, which is going to give us the link between inflammation and adaptive immunity. And the key thing to understand, of course, conceptually, is that in order to activate adaptive immunity, we need both something to be foreign, to be recognized, and then we also need to have a signal that tells the T cell, if it's starting with a T cell in the lymph node, that this dendritic cell has come from a land of danger. So if the dendritic cell is activated by a TLR, it will have the ability to provide a second signal. So normally, let's assume you had a foreign protein, got taken up by a DC. Maybe there's a peptide, this brown peptide presented on class 2. It could be recognized by the T cell receptor. This is foreign, and it can only provide signal 1. This is not enough to activate a T cell. We always need a second signal. So TLR activation will induce a co-stimulatory ligand like B7. B7 will trigger CD28 on the T cell. So this is the naive T cell in the lymph node. And now we will have activation of the naive T cell in the lymph node. Okay? The other thing we want to remember, of course, is that when a cell captures, a dendritic cell captures a pathogen antigen and then gets activated through TLRs, it migrates through afferent lymphatics to the lymph node, provides these two signals, one for foreignness, one for danger, activates T cells, and these T cells now become effectors. And it is the effector T cells which are going to go and migrate back to the site of inflammation and deal with the pathogen. And these effectors, some of them will become memory cells, the majority of them will die, and that's called contraction of the immune response. So we make immune cells in central lymphoid organs. We generate diversity of the repertoire in central lymphoid organs. We have central tolerance in central lymphoid organs. Naive cells only go to lymph nodes and to spleen and Peyer's patches, so secondary lymphoid organs. That is where naive cells get activated, and they need to get activated by two signals. Okay, and then these naive cells got activated. They migrated back, went to the site of infection as effectors, dealt with the pathogen, and then maybe some of them become memory cells. And then again, just broadly speaking, I'm actually going to talk about a CD4 subset in the context of disease, where I try to explain approaches to human immunology. I just want to tell you that the classical thinking, even in our textbooks, is that we have Th1s, Th2s, Th17s, TFH, Tregs, and if Amit is in the room, always Th9 as well. But there are some more subsets to think of. And certainly today, I'll talk a little bit about the one that might be linked strongly to disease, about which we're learning more. Okay, so if you think about disease and you think about how we consider disease and how we divide disease in general, there's a few categories we can break everything into. Okay, so we have Mendelian disorders, single gene disorders, relatively easy to understand. Infectious diseases, even COVID-19, again, you have some combination, like if you now know the story about type 1 interferon receptor pathway mutations, there can be a combination of genetics and a microbe which can cause a disease, and this would be a severe infectious disease. In cancer, again, relatively easy to understand, not that we can solve everything about it, is that a single cell acquires somatic mutations, and those somatic mutations allow the cell to grow in an uncontrolled manner. And everything else, which is the real frontier today, both in biology and medicine, is a complex disease. And complex disease is something that everybody on this Zoom call has in some form or the other. If they don't already have it, they'll have it in the next 10 years or so. And this includes type 2 diabetes, atherosclerosis, hypertension, asthma, allergies, autoimmunity. All of them are complex diseases. And we hand wave and call them multi-gene disorders. We don't fully understand them. Now, there are some occasional diseases, and these are 2 occasional tumors, where we actually have no driver mutation, but we have epigenetic changes. And these somehow will fit into the same category as complex diseases, okay? Because there's a lot of epigenetic alterations driving disease. So what do we think is happening in a complex disease? And again, I have to say, we don't know all the answers yet. Obviously, this is a frontier. There's some genetic susceptibility. There's no question there's some amount of it. It cannot explain everything. There's usually a trigger. Now, if this is not an infectious disease, then there still may be some change in the microbiome, some metabolites generated, and these are being studied. And there's very interesting data coming out. And even in the disease we study, we collaborate closely with Ramik Xavier to look at the microbiome. So either antigenicity shared with the microbe or a novel metabolite coming from a microbe might cause an epigenetic alteration in an immune cell. Then some clone of immune cells, which is altered, might then break tolerance and lead to enhanced inflammation, and this might be the cause of a disease. Okay, this is the way we think about it. And clearly, when we study COVID-19, we're going to be following patients because now we have an idea of what the trigger was, and then we want to see what are the consequences. The other point that I think we need to understand, when we study disease, we do have to study disease not just in the blood. Unless you're studying, just a leukemia, which is highly bloodborne, you do need to go into a couple of other sites. So if we want to analyze a human disease, one thing we have to do is to look at lymph nodes. And lymph nodes, as you know, have both T cell zones and B cell zones. And we need to be looking at lymph nodes in the context of disease, because the immune response that causes a disease is likely to arise from a draining lymph node. You might have factors in the tissue, but then the draining lymph node is important. And the other thing you want to consider, of course, is that there's a lot of things that happen only in lymph nodes. For when I talk tomorrow, we cannot look at germinal centers in the blood. You have to look at a lymph node. And I would say one step further, in any disease which involves an organ, you need to look at the organ as well. So to be comprehensive in the immunological approach to human disease, we need to be looking at many things from genetics to microbiome and so on. But we need to interrogate the blood. We need to interrogate lymph nodes when we can. And we need to interrogate the tissue, the organ in which the lesion occurs, because then we are going to get a better picture. And what I'll do today is we study a few diseases in my lab. So we study systemic sclerosis, we study IgG4-related disease, we study fibrosing mediastinnitis. This disease is actually caused by Histoplasma capsulatum. And it gives us some clues because we know there's a microbial cause to fibrosis. These are all fibrotic diseases. We actually look at some diseases in which there are single gene mutations. So we have a huge cohort which we have built together at Mass General, about 270 patients now, of patients with single gene mutations in the immune system. But we are focusing on those that cause autoimmunity, fibrosis, and which block B cell development. And we have some interesting stories. I won't have time today to get into that. And I'll talk about COVID-19 tomorrow. So today, I'll largely be focusing on the first two as examples to explain strategies for how you approach a human disease. So we think about the workflow for studying human disease. When you can interrogate blood. So, one standard approach we like to use is 25 color flow panel for T cells, 25 color flow panel for B cells. This allows you to purify cells, do RNA-seq, ATAC-seq, cut and tag. Single cell RNA-seq can be done from the blood. You can look at repertoire. Some people prefer to do cytoff as the first thing. We reserve it for certain reasons. We can do antigen discovery. You can look for what is the antigen that in a disease a B or a T cell sees. And a tool that we've started to use a lot more with Willie Haas at Mass General is mass spectrometry. We can get 8,000 proteins easily from a specific immune cell. And then, so mass spectrometry is again unbiased. It's like single cell RNA-seq, but looking at proteins. And at the end of the day, you want to look at proteins. We can interrogate draining lymph nodes, and we prefer to use multispectral imaging approaches. We can do cell distance mapping there, but you can also use Hyperion, you can use Codex. There are other alternatives that some people might prefer. I just think that morphology-wise, we do better with what we're doing. We can interrogate the target organ. So if I have a disease in which the skin is affected or the lung is affected, or maybe the pancreas is affected, I can go and look at those tissues. Again, we prefer to use this multispectral immunofluorescence, and you'll see more of it tomorrow when we do lymph node studies. But I would like to do cell distance mapping. We want to know which cell is talking to which cell. What is it making? And what's the frequency of that interaction to quantitate it. Single cell RNA-seq doesn't tell us that because you broke everything apart, but we like to use this as well. But you can use, obviously we use single cell RNA-seq as well. It's a powerful tool. Again, you can use other tools. Slide-seq is another tool that hasn't actually worked out as well as advertised. Then we need to look at underlying mechanisms. What's the microbiome like? We do antigen discovery as mentioned above. You can look at the chromatin state of cells that are driving a disease, see what epigenetic alterations drive those cells. Then you can test the validity of any model you build. For this, you have to do clinical trials as one approach. Sometimes that will give you a clue. If you can, you model it in mice or in humanized mice. Okay? These are and you can use CRISPR, of course, to do a lot of the modeling. So what do we think you need? And if your disease is caused by T cells, we think there are some Cox postulates we can use. Are there clonal expansions of effector memory cells? If there are clonal expansions, then we can look at them in detail, and you can look at, prove it's a clonal expansion by next generation sequencing. I'll show you an example of that. If you've identified a T cell clone in the blood and you think it's expanded, is it in the tissue? If it's not in the tissue, what does it mean, right? It has to be, if it's causing the disease, it should be in the tissue where the lesion of the disease is. So very important to go look at the tissue. And then if you think that clone is driving disease, or those clones are driving disease, you need to look if therapy, any therapy that improves the disease, that it cause the loss of those clones. So if you fulfill these postulates, you have a better idea of what's going on. So I just want to put it out there. We're looking at a few fibrotic diseases. A lot of people die from fibrosis. There's so many different causes of fibrotic diseases. I'm going to first give you the broad model so that you can follow along, and then I'll talk about a couple of diseases with examples of how we interrogate disease. So this is our broad model, and it applies to 2 or 3 diseases now, at least 3 that we've looked at. And we think CD4 CTLs and sometimes CD8 cells are driving the disease. They are clonally expanded, and I'll explain what CD4 CTLs are in a bit. We think activated B cells are playing a role. They're contributing. Maybe they're presenting antigen. Maybe they're making things. I'll show you they can make enzymes. You would never believe that B cells are doing these weird things, but they do. We find some of these weird B cells in COVID. We just published that recently. I'll talk about briefly tomorrow. We think you then have the induction of apoptosis. So we think in every severe fibrotic disease, even in pulmonary fibrosis, which can have genetic causes, you have apoptosis. And some target cell is dying by apoptosis. And then when it's remodeled, space is created, you remodel, you over-aggressively remodel, you get fibrosis. So you have macrophages, fibroblasts, myofibroblasts all involved. But we think the main trigger here is immune-mediated apoptosis, setting the stage for now to replace those dying cells with a fibrotic tissue, often a fibrotic mass. So systemic sclerosis or scleroderma is a very bad disease. It reduces the lifespan of women who have it by 20, 25 years sometimes. And it occurs predominantly in women of childbearing age. If you see a patient, it's a very severe disease with affecting almost every organ you think of. But we worry most about things like the lung, the kidneys, the gut, and the skin. Okay. Now, there are endothelial alterations in this disease. And there's always been a belief there's some connection to the endothelium. And I'll tell you what we think is going on. The other disease that I'm going to use in parallel, because it's almost like a model system to go and study systemic sclerosis, is IgG4-related disease. Again, here you have a mass. Patient often presents misdiagnosed as having a tumor. It's actually just a mass. In that mass, it's inflammatory cells that have collected. You see blood vessels are occluded. It's called obliterative phlebitis. And you see storiform fibrosis. It's just a description for the type of fibrosis. For diagnosis, typically you have to say very high proportion of the cells, the plasma cells in the tissue, are plasmablasts. But the majority of the cells are actually T cells. You have T cells and plasmablasts, activated B cells of various sorts that we have looked at, all in the tissue. And it can affect almost any organ. So I'm just going back to some work we did when we started to look at IgG4-related disease, looking at the blood. We gated on effectors, and we found, okay, yes, in the disease, there's lots of effectors here. And of course, in controls, effectors are a very small subset. You can't see them. We then looked at these effectors and characterized them transcriptomically and found that they're kind of weird, that these effectors all had a signature of a CD8 T cell. This is a CD4 effector. It has a CD8-like signature. And these cells have been described in different contexts, but I think we established that you can have disease because of these cells. And these cells express TBET, CD97, SLAMF7. They've turned on a program of CD8 cells. And I'll tell you towards the end how we think this has happened. Okay? So there's CD8 programs turned on CD4 cells. These cells are called CD4 CTLs. They protect against dengue. That's well described. They protect against other viruses, including CMV. They're induced in HIV, provide some protection, obviously not enough. And they also have been shown in cancer. And so tumor immunologists are now very interested. So both Jim Allison and Jed Walchuk have papers on CD4 CTLs in cancer. But we've looked at it in inflammatory disease. The numbers of CD4 CTLs in active disease goes up dramatically compared, this is all blood, compared to controls. Okay? So we asked, are these clonally expanded? And the way you look at it is you take, let's say you took out 50,000 cells and you want to ask, is this clonally expanded? You take those cells and we have rearrangement of the beta chain and alpha chain of the T cell receptor in a T cell. So we basically do next-gen sequencing, and we'll may sequence 5 million times to ask, is there a clonal expansion? Are all these cells derived from 50,000 different cells, all the sequences? Or they derived from 10 different cells or 50 cells? What is the answer? So when we did that, so you can see this, these are 4 patients with, this is the one part of a study we did where we chose people. So remember, about 40% of all humans, or 30 to 40% have allergy. So we chose 4 patients with an IgG4-related disease with a history of allergy because we can't find Th2 cells in the blood otherwise. We wanted a control. So these are patients who have got IgG4-related disease and they have allergy. We can purify the Th2 cells. This is the GATA3 positive cells. Those are the SLAMF7 positive CD4 CTLs. And what we find is CD4 CTLs are big clones. Whereas this is the history of allergy in this patient, the little dots. Okay, so there are big clonal expansions of CD4 CTLs in this disease. In the tissues, we can find these cells in the tissues. You can quantitate them. This is in a bunch of patients. And you can see that the dominant CD4 cell here is this CD4-CTL, and Th2s are relatively rare. Okay? In systemic sclerosis, given that there's a highly fibrotic disease, we did studies in parallel, and this is again something that was published earlier this year. And here we showed that in systemic sclerosis, of course, there's lots of CD4 positive, granzyme A positive cells, and very few GATA3 positive cells. On the other hand, if you look at bullous pemphigoid, which is really a Th2 dependent disease, there's lots of GATA3 positive cells, and they are Th2s. So if you quantitate again, the red bars are CD4-CTLs, the green bars are Th2 cells. So let me go back. And you can see that by and large, there are a few patients in whom, there are a few out of these 35 that we're looking at, there are a few in whom you do have good Th2 cells. But for the vast majority, when we quantitate it, the majority of the cells, the dominant cells are CD4-CTLs. This is different in bullous pemphigoid, where the dominant cell is the Th2 cell. And this is the quantitation over here. So in systemic sclerosis, you tend to have more CD4-CTLs than in bullous pemphigoid. We took 2 skin diseases, so we could do these comparisons. If we look at all CD4 T cell subsets, so we are able to stain all the cells in the same experiments, and we quantitate CD4-CTL, Th1s, Th2s, Th17s using different transcription factors, TFH and so on, and Tregs. You can see CD4-CTLs tend to dominate. So just by using quantitation, and as I'll show you in a minute, by looking at actual cell-cell interactions, we can make some conclusions. Say, yeah, these cells are the dominant cells, and they're in the tissue. And this is not in every disease. In certain diseases, the ones we're looking at, this is dominant. Then the question was, do these cells induce apoptotic death? And then this interesting result came out. We noticed that endothelial cells are undergoing apoptosis quite a bit in this disease, in systemic sclerosis. In IgG4-related disease, we have apoptosis, but not so much of endothelial cells. So these CD31 positive cells are endothelial cells. And you can see cleaved caspase 3 inside these endothelial cells. And the blood vessel cells, some cells are undergoing apoptosis. This is, I'm not sure how well you can visualize this, but here's an example of a CD4-CTL, which is granzyme A positive. Okay. It is actually in the vicinity of a cell over here, which is essentially this endothelial cell. This is the endothelium. But that endothelial cell has acquired a little granzyme A. So you believe the granzyme A has been delivered by the CD4-CTL into this endothelial cell. And that endothelial cell is undergoing, it's got cleaved caspase 3, it's undergoing apoptosis. And we've done this a few times. So I think we're even able to capture, if this is the endothelial cell here, which is dying of apoptosis. And this is the endothelium. You can see the circular form here. And the CD4-CTL has apparently executed it by putting granzyme A into it, and it now has cleaved caspase 3. So again, by, we can use tools like cell distance mapping software, ask which cell is kissing which cell, quantitate it, and so on. So we see these interactions, strong interactions between CD4-CTLs and endothelial cells, and frequently apoptosis of the endothelial cell. And then we can quantitate it and say, this is what's commonly happening. So then if I give you a model for systemic sclerosis, the model says that cytotoxic cells, mainly CD4-CTLs, may induce perforin granzymes, target some antigen, obviously very important to know what the antigen is, in an endothelial cell, and the endothelial cells now undergo apoptosis. In this context, there is some role, we think, for activated B cells in the process. And there are now some papers, even one from India, showing that rituximab is improving a little bit the outcome in systemic sclerosis, not a huge difference, not like IgG4-related disease. So there may be a role for B cells in the process. And at the end of the day, you will now get fibrosis. Okay, and this is fibroblasts and macrophages play a role at the end stages. But I think the key event now is immune recognition of an endothelial cell by a CD4-CTL. Okay? HLA-DR is expressed at high level in these endothelial cells. So during inflammation, this is what happens. So this also explains, so I didn't show you that for systemic sclerosis. I'm now showing you this for IgG4-related disease, where in inflammation, HLA-DR gets expressed on many cell types, but in IgG4-related disease, endothelial cells are not dying, but Vimentin expressing mesenchymal cells are dying. So whatever the driving antigen is in IgG4-related disease, not the same as systemic sclerosis, though the mechanism may be similar. Okay? Again, we have quantitation over here, and we're comparing Sjogren's syndrome here with IgG4-related disease, and you have a fair amount of mesenchymal cell apoptosis in IgG4-related disease. So a broad view now, obviously this is all human. We are based on comparisons, there's some heterogeneity, but we think systemic sclerosis is a CD4-CTL driven fibrotic disease in which the antigens are endothelial. IgG4-related disease is also CD4-CTL driven disease in which the antigens are stromals in fibroblasts and myofibroblasts. In fibrosing mediastinitis, we have actually shown this paper has already been accepted, it's not out yet, that the CD4-CTLs that are maybe driving the fibrosis, we can't prove that, but these CD4-CTLs are specific for histoplasma capsulatum, that we've shown. Okay? So in some cases, it may be an autoimmune fibrosis, in some cases, it may be pathogen-induced fibrosis, but CD4-CTLs may be playing a role in the disease. Okay? Now, I said a lot about CD4-CTLs. How do we study these cells more? How do we put it all together? So we have been using epigenetic approaches and single-cell RNA-seq and other tools, which I'll show you a little bit about, to understand novel disease-related human immune cells. And if you get enough cells, you can do a lot of studies, and some of these tools can be used with very few cells. So most of you have heard of single-cell RNA-seq. I won't belabor this. You can use single-cell RNA-seq. You put one cell per droplet or one cell per well, get transcriptome data, and that's your single-cell data. Okay, and a lot of computation, not very complicated, but a little computational work to resolve it. This is some of our data. This is published now using single-cell RNA-seq, repertoire, flow cytometry to look at subsets of CD4-CTLs. And we can divide this into subsets. And the subset that causes disease and which expands and clonally expands is the CD4-CTL effector subset. We call it effector because we think it's driving disease. But there is a number of, there are three different subsets we can see by single-cell RNA-seq. Again, here you'll see by single-cell RNA-seq, this is just using time of flight. There are three different subsets. We put it in this context. This is actually by flow. We are looking at the subsets because you can actually characterize them, and then we can look at them in much more detail. We've done RNA-seq on them, looked at what's different between subsets, characterized these differences, and clearly the ones that we call effectors are the ones that we think are driving disease. We've correlated with disease. Now, none of your studies are worthwhile. None of our studies are worthwhile unless you have a good clinical correlations, good characterization of patients, subclassification, and we do all that to try to understand it. This is to show clonal expansion, which ones are highly clonally expanded, and you can see that over here, that the frequency, these are the cells in which you have the frequency of clones which are going, what is the top 10 clones in each subject, okay? This is what we see. Okay, how about epigenetic marks? So the way to think about this is, of course, that if a gene is going to be expressed, it's in what you would call in heterochromatin or in a closed chromatin state. When a gene is, when it's not expressed. When it is expressed, you're going to have movement. You're going to have opening up of, so the nucleosomes are going to move apart, and you're now going to expose the gene to be expressed. Okay, so this is just a simplistic way of thinking about it. But one way to think about gene expression in a specific immune cell, and this has come from many people's work in the past, is that if you look for genes which are driving the biology of that cell, they will be regulated by regulatory elements we call super enhancers. And a super enhancer is nothing but the fact that you have just defined by size, how many binding sites are there for many different transcription factors. So a typical enhancer will have a few sites. A super enhancer will have many sites. It'll be a larger piece of open regulatory regions of a gene when it's expressed. This is the transcription start site shown over here. So if you can identify which are the genes in which there are super enhancers, that already gives you a clue which are the genes that drive the fate of that cell and drive the biology of that cell. It's just a tool to use. And basically, this is a slide saying similar thing that, typical enhancer, you form this structure over here. It's a TAD, or a transcriptionally active domain. And in the case of super enhancer, you have a lot of binding sites, a lot of transcription factors, a lot of protein-protein interactions driving this. And this is why this gene is expressed so much better in that particular cell type. So we use this as a tool. I'm not going into the methodologies of these. We could discuss that, but we use things like ATAC-seq, we use DNA methylation in order basically to identify super enhancers. We use ATAC-seq and we use histone modifications. Anyway, so we identify genes linked to super enhancers in CD4 serials and some TFH subsets we're interested in. This helps us identify pathways and enzymes. Then the whole goal is, if we know more about this, then we can generate small molecule inhibitors and maybe new drugs to suppress immunity. Now, I'm going to just show you a little bit of our data. So this is, if we're looking for super enhancers, and we're looking in both CD4 CTLs and TFH cells. And in TFH cells, BCL6 comes up on top, which you will understand makes a lot of sense because BCL6 is the master driver of this cell. And in CD4 CTLs, we come up with multiple things. And so these are the things we're investigating. This is just looking at differential exposure. We're looking at naive cells, TFH cells, CD4 CTLs. Just by looking at this, you can tell there's differences in what we are defining as our super enhancers in these different cell types. The most interesting thing that we found about CD4 CTLs is that if you look at any lineage of T helper cells, they are slightly demethylated compared to naive cells. But when you look at CD4 CTLs, we're now comparing the degree of demethylation. So here are our two controls. This is showing you one example here. This is the degree of methylation, whereas the CD4 CTL is dramatically reduced. It goes down to about 65% of methyl CpG compared to naive. Methylation usually is a simplistic way to say it is, if the gene is highly methylated, it's not going to be expressed. If it's demethylated, it's going to be expressed. So just to show you a couple of drivers. So RUNX3, important transcription factor for CD4 CTLs. You can see these two lanes are showing you CD4 CTLs. You can already see open chromatin here, which is not seen in naive cells. Let's show you one more example. If you think of perforin, crucial in CD4 CTLs and CD8 cells, open chromatin. So you had to demethylate heavily during development. You had to take a CD4 cell, it went through a program, you demethylated it heavily in order to now get the expression of this new cassette of genes, which are making it more like a CD8 cell. This is the naive cell, this is the CD4 CTL. Obviously, I just showed you a couple of examples. The other question you should ask is, did therapy lead to attenuation of CD4 CTLs? So, if you got the disease to improve, do CD4 CTLs decline? And so in IgG4-related disease, we had done this. So here we treat patients with rituximab. This is just depleting B cells. And you can see clinical outcome improves dramatically with treatment. IgG4 levels don't drop that much. They drop a bit. B cell depletion is perfect. You can expect that. Plasmablasts do not have CD20. So rituximab targets CD20. But they decline after time because they have to come from B cells. They have to be B cells have to be activated. So if you look after a month, they've started to decline quite a bit. CD4 CTL, the effector memories drop. CD4 CTL clones, we can follow specific clones because we know the V beta sequence. They also decline. Okay, so there is a decline in the CD4 CTLs when treatment works and patients get better. So this has led us, and I'll only touch on this again to think about methodology. How do you approach things? So B cells, what role are they playing in fibrosis? If you depleted B cells, disease got better. Is there anything they do beyond presenting antigen to a CD4 CTL? So I just show you this to show you an example, just using t-SNE plots. And this is we do 25 color flow, but this is using a few colors to compare and paint the cells. If you look at the control, and then you look at somebody with IgG4-related disease, and this is from about 40 patients, we've got similar results. You can see that there are some new islands over here, which did not exist in the control. You can see the other islands are virtually identical. Okay, everything looks similar. You just look at the patterns. they're almost identical. But these islands here, these ones with arrows, do not exist in the control. So novel cells are being created in a disease context. We now understand that all of these cells are created outside the germinal center in an extrafollicular site. And I'll show you some of that data in a COVID context tomorrow. These cells, some of them are secreting lysyl oxidase 2. Okay, and lysyl oxidase 2 can cross-link collagen. This is to show you that, yes, in the disease tissue, we have cells that are making IgG4 and are also making lysyl oxidase 2, and that this is something that goes on. Can we prove it in a human that it's important? No, it's hard to prove. We can only quantitate it. But so that's why we always need to go back to animal models to prove mechanism, which is something that we cannot do just by looking at humans. Okay? So again, the big picture view, we come back to this. This is the pathogens of autoimmune fibrotic diseases. You have CD4 CTLs, you have activated B cells, they may work together, and you get cell death. Okay? So I'll spend a few minutes talking about antigen discovery. Obviously, you want to know what antigen are the activated B cells seeing and what antigen activated T cells seeing. Are they the same antigens? And T cell antigen discovery is more difficult, but we are doing it now. I'll mention it at the end. If you're interested, I do have some slides on how it's done. So for B cell antigen discovery, pretty easy. B cell antigen discovery, what we do is we make monoclonal antibodies. And this is what's same thing was done by the way in COVID, the similar approach, what I'm showing you here. So you take a patient, you take the plasmablasts, he's got active disease, you put one cell per well, okay? And then you, from each cell, you pull out the antibody heavy chain and light chain gene. Okay, then you put these into cassettes where you can have the rest of the antibody heavy chain and light chain. You PCR it out, put it into a cassette, and you make a monoclonal antibody. It's a human monoclonal antibody, one coming from one from each cell. And this is what is done. But the antibodies that Trump received, I can't dignify him in many ways, but he basically received 2 monoclonal antibodies derived in this fashion from a COVID-19 patient. Okay, well characterized. Anyway, so these antibodies in this case were staining some cells. It was interesting. This is showing you that by next-gen sequencing, these peaks came out to be the same as what we picked by single cell. So the ones that are more commonly as single cells were also more common by next-gen sequencing because, can actually sample more cells. And then we stained pancreatic cells and shown that, yes, these antibodies are staining some antigen in pancreatic cells. So then it's easy to try to purify it. Fairly simple to take the antibody, conjugate it to cephros, make a lysate of pancreatic cells as a source of antigen, do affinity chromatography, do mass spec. And this is what the mass spec shows us. There's always some noise, but you can see in all 3 clones that we pulled, the peak antigen picked up was galectin-3. So we now know in a subset, it's only about 1/4 of patients or 1/3 of patients with IgG4-related disease, galectin-3 is an antigen. In some other patients, we did with Bill Robinson's lab, we used microarrays looking for anti-cytokine antibodies. She found a very interesting anti-cytokine antibody, which is against the IL-1 receptor antagonist. Might explain why the disease gets worse because IL-1 is being made by CD4 CTLs as well. So that's something else, another approach that we've used. There are other approaches like Plato that we've used and so on. And so there are multiple approaches that can be used for B-cell antigen discovery. For T-cell antigen discovery, which is what we're doing right now with Michael Birnbaum, who's our neighbor, he's at MIT actually, but he's next door. We are the Ragon Institute is next to MIT actually. So we take, in this, what you do is, so single cell RNA sequencing, and we do TCR sequencing, so on those single cells, and identify clonal expansions, and then we make soluble T-cell receptors. So you made the T-cell receptor from the biggest clones. But you predict which is the relevant HLA by clustering patients. So let's say I have 50 patients, and 10 of them had the same T-cell receptor, V beta usage, or V beta plus J beta usage, maybe the same TCR alpha usage. So they have very similar T-cell receptors. Those who share T-cell receptors must share HLA. I mean, that's the assumption we make. And this method was really derived by Mark Davis called Glyph. So we worked with Mark Davis on this. So then you try to predict the relevant HLA by doing this. This is actually done quite successfully by us. I don't believe it will work every time. It works sometimes. So this works, at least in this disease. Then you take, this is again done by Michael Birnbaum, you take the library of all human peptides, all known human peptides, and attach it to the HLA of choice that you discovered by glyph. And then you screen a library and ask, the soluble TCR from that big clone, does it recognize which peptide does it recognize? And thus you identify the peptide. Now, I do have slides. If you're very interested, I'll go to those slides, but I'm going to close with a summary of an overview of immunology, but it'll be in a different sort of presentation form. I just want to mention that a lot of people in my lab were involved in this work. Almost all of them are involved in various aspects of our work, whether it is on systemic sclerosis or COVID-19 or IgG4-related disease or on the single gene mutations. There are some ex-members. There's actually one person who was at NII many years ago who was a key person in my lab. That's Hamid Mattu. Vinay Mahajan is actually a graduate of All India Institute of Medical Sciences and did his PhD at MIT after finishing medical school there. And there are others who are very important. I have key collaborators. John Stone is my key collaborator at MGH. For all of we do clinical studies, clinical trials. I mean, I can easily convince him to do a clinical trial. There are many fellows in my lab who are shared between, who actually go to the clinic and also come and work in the lab. Seiji Nakamura has been helpful in Japan with a lot of clinical material. Dinesh Khanna is my key collaborator in systemic sclerosis. He's at the University of Michigan. For those of you who are interested in Bollywood, he's closely connected to an ex-Bollywood person. I won't name him right here. And there are other people who are involved. Ramnik Xavier for microbiome, I didn't talk about it. Alex Shelik has helped us a lot with single-cell RNA-seq. And then I didn't talk about a single gene mutation project where we have other collaborators. So I'm going to end with the lymphocyte wrap. I think I'll stop sharing and I'll do this. Yeah. And this is just to summarize the whole, approaches to immunology. I have to warn you that unfortunately, this involves my moving my hips. You are not going to be able to see that. My hips are moving. I'm sitting on a chair. Just know that my hips are moving while I do this.
Thank you.
Pathogens lurking everywhere.
Yeah.
Go get them first. They never fight fair. Give me a D, give me a J, put a V in frame. It'll be A-OK. Surrogate light chains, pre-TCR, kappa and alpha can't be too far. Thread that peptide into TAP. Everybody do the lymphocyte wrap. Naive cells dance from hall to hall, pining and hoping to get that call, looking for antigen below and above. Many will die of unrequited love. Thread that peptide into TAP. Everybody do the lymphocyte wrap. DC's lurking everywhere. They'll get your antigen, don't despair. CCL21 to CCR7. Come in, DC baby, make this lymphocyte heaven. Thread that peptide into TAP. Everybody do the lymphocyte wrap. Wake up, T-cell. Activate ZAP. T-cell says, takes 2 hands to clap. Give me signal 1, give me signal 2, and I'll give you a cytokine brew. Thread that peptide into TAP. Everybody do the lymphocyte wrap. Collectins, complement, macrophages. That's what you get in the early stages. In antibodies, cytokines, CTLs too. Hey, pathogen, that's the end of you. Thread that peptide into TAP. Everybody do the lymphocyte wrap. Thank you for listening. I'm done. I can take questions.
Thank you.